Miles, Dennis, thank you very much indeed. We will now, on a completely different topic, to some extent, I think, travel back into the past. Richard Padley, how DNA has relevance beyond dinosaurs. Richard. Richard, you also need to unmute yourself, please. We're on fire with this today.
There we go. Thank you, Andrew. When Dennis mentioned a few moments ago, people thinking of a quartet and Led Zeppelin songs, can I counter that with a slightly different quartet, which is, of course, the 4 Jurassic Park films. And that's probably a slightly bigger quartet than Led Zeppelin ever were. Now, when many people of my generation first think of the concept of DNA, the film Jurassic Park comes to mind, because, of course, for Milo and Dennis's benefit, that was a film that used DNA from mosquitoes, that it extracted it and brought dinosaurs back to life through some sort of clever forensic process. And of course, when jurors walk into court and hear about Jurassic Park, well, more likely to hear about DNA than Jurassic Park, their minds straight away go to these sort of crime forensic programs, CSI and the like. And they think the DNA evidence is very strong. Well, the purpose very much of this talk is to give you a bit of ammunition as to how to challenge it and how it can be questioned. Now, let's go, please, Andrew, to the first slide, and we'll start off with a little bit of science. Now, the DNA of relevance to forensic science that's used in the criminal justice system is found in the nucleus of the cell. Whilst DNA can also be found in mitochondrial DNA, it's the nucleus and genomic DNA that we're going to focus on. DNA, of course, provides a complete genetic code for a person, containing a record of each person's individual features. Whilst it was originally considered back in 1869 by someone called Johann Miescher, it wasn't until 1953 when James Watson and Francis Crick revealed the structure of the DNA, and they provided an explanation for how the genetic information can be passed from generation to generation. But it wasn't until the 1980s when research was undertaken by Dr. Alec Jeffreys that demonstrated how DNA contained patterns and how those patterns varied between individuals, and importantly, how that could be used in the criminal justice system. Well, DNA, much like the image you see on the left side of that slide, is presented in a form of a double helix ladder type structure. But if we unwind that ladder, what we get is a series of rungs, each containing 4 separate chemical bases: adenine, thymine, guanine, and cytosine. A, T, C, and G, you'll sometimes see them referred to. Now, those 4 base pairs match up in a particular manner: adenine with thymine, guanine with cytosine, AT, G with C. And it's a subsequent order up and down the ladder of those base pairs that give DNA its unique profile, and us as humans, our own unique profiles. Now, within the human DNA, there are approximately 3 billion DNA base pairs, as was seen and as was quoted in the 1993 film Jurassic Park. But let's step back a little bit and consider these matters on a slightly bigger scale. I say bigger scale. Let's go to the nucleus of a cell and consider where the cell genomic DNA arranges itself into the form of chromosomes. Yeah, thank you, Andrew. Still on that slide for the moment. All body cells, with the exception of sex cells, contain 23 separate pairs of chromosomes, so 46 in total. We have 23 from the male and 23 from the female that have come together. The only exception to that rule is sex cells. Sex cells, sperm cells in men, egg cells in women, contain 23 chromosomes. And when the egg is fertilized by sperm, the resulted zygote cell contains a fused 46 chromosomes. So a little bit of science there to keep us all going at 5 to 4 on a Thursday afternoon. Now, each chromosome has a number 1 to 22, and then the sex chromosome X or Y. A male will have 2 X sex chromosomes, female an X and a Y. Now, on a chromosome, there is a locus. A locus is what's known as sometimes referred to as a DNA marker, and that you may see referenced in forensic science reports. That's the place on a chromosome where a particular gene can be found, and a gene is a specific sequence of DNA molecules. Within the gene, you find a series of alleles. Each individual will inherit an allele from the father, an allele from the mother, and it's the comparison of these that the forensic scientist will ultimately look at. Within the allele, you get a sequence of those A, T, C, and G base pairs. Can we now move on to the next slide, please, Andrew? Now, the development of how DNA has been used in the criminal justice system started off with the first generation multiplex right at the bottom there, and leading up to what we presently have, the DNA 17, which is a 16+1 system. Now, what that means is that the forensic scientists will look at 16 separate individual locations, 16 locus, plus the sex locus, when it comes to consider whether a comparison between two sources of DNA can be made. That system was brought in over the second generation multiplex system. That was introduced in 1999. That considered, as it says on the slide there, 10+1. The second generation multiplex considered 6 locus locations, with the first generation multiplex considering 4. Can we move on, please, Andrew? Sources of DNA. What potential sources, when a crime scene investigator turns up at the crime scene, what can they look for to consider, well, are we going to find DNA in this item? Blood, yes. It's not the red blood cells, though, because red blood cells solely carry oxygen and nutrients around the body. It's the white blood cells. And where those cells are located can be quite important, because when a person is cut and when they first start to bleed, it's the body's first line of defence to try and stop that bleeding. And it does that by flushing out a series of white blood cells to the wound with the aim of stopping the bleeding as quickly as possible. Now, of course, those white blood cells contain a very rich amount of DNA. And so when DNA and a blood source is considered that's close to the scene of where the cut has taken place, you're going to get a better quantity of DNA, which is more likely to produce a result. Semen DNA material is located in the spermatoza, and it's possible to identify whether a sample of DNA is in fact semen through the use of a seminal acid phosphate or a protein specific antigen. Again, all fun for 4 o'clock on a Thursday afternoon. Saliva. Saliva can be found on items such as bottle tops to the left at the scene. It's not the saliva itself that contains DNA, but when, for example, somebody takes a sip of a drink, it's the saliva that transfers from the inside of the mouth, carrying buccal cells that will have been transferred to the item. But it's worth bearing in mind when taking a DNA sample from a bottle, for example, acidic and sugary drinks can have the effect of degrading the quality of the sample. So it may not be that the result that's obtained is particularly good. Sweat doesn't itself contain material in a DNA material in a pure form, but just as with saliva, if it's come into contact with skin, it may well be that some of the skin samples have taken DNA off with it. And finally, mucus we have there. Well, mucus, again, if somebody is sneezing into a handkerchief, there's a lot of force behind that. And the DNA can come out when the mucus touches the skin, resulting in DNA being present in mucus. Can we have the next slide, please, Andrew? Now, some big words on this slide. More fun and games. How is DNA in fact obtained? Well, there's a number of stages which the sample will go through to extract the DNA, to put it in a form that it can be assessed and analysed in an expert's report. The first is a process of extraction and purification. This is when the sample is checked for DNA cells, which involves disrupting the cells that have been obtained, removing the non-nucleic components of the cell, which leaves solely the nucleic part which contains the DNA. We then have a process of quantification. The DNA that's been extracted is quantified, it's measured in terms of the number of base pairs that have been obtained, and two relatively big words there, spectrophotometry and spectrofluorimetry are two processes by which quantification can take place. Which one is used ultimately depends on whether the sample is a high level sample or a lower level sample, but there's ways in which it can be obtained. Amplification, a polymerase chain reaction. If the DNA sample that's being obtained is good enough, this polymerase chain reaction is a process that involves repeatedly heating and cooling and stretching the sample over a number of cycles so that the DNA fragments increase in size. It's a process of effectively multiplying that sample time and time and time again so that it's at a size that can be seen and assessed. Finally, detection and interpretation. The DNA sample is then passed through a laser, causing chemical fragments to fluoresce, allowing them to be detected and recorded on a computer, at which point the genetic mapping and sequencing takes place. Now, that of course is all done through a computer generated piece of software. How peaks are determined, how high they are is determined by the piece of software, and of course the underlying computer code. At this stage, we've not necessarily got to a significant amount of input by a forensic scientist interpreting the results. A lot of this is done by a computer piece of software. Now, you'll recall from the seminar that I gave a couple of weeks ago, Section 30 of the 87 Criminal Justice Act, potentially the 88 Criminal Justice Act, which allows the prosecution to rely upon the work of others when performing this analysis and providing their interpretation. Well, here, the forensic scientist is relying upon the computer code. It's the computer that has performed the graphs, it's the computer that's performed the analysis, and it will be the forensic scientist that comes in to analyse those and brings them together. So there's potential issues of disclosure that arise at this stage. How has the computer software done its job? It may well be that one thing you choose to do is go to court and get that Section 30 order that says the prosecution can't rely on the underlying work of the software as a way of challenging the potential calculations that have been done. Now, let's move on to the next slide, AJ, and we will get on to electropherograms, helpfully hyphenated to assist me with the pronunciation of this word. The final stage of this process of extraction of DNA involves looking at these bars that we see. And what we have on this particular graph are a series of locus points that have been identified as part of the expert's report. And then a series of bars of varying heights. And this is known as the electropherogram. And this is what the forensic scientists will be looking at. And it'll be very much a case of comparing the electropherogram that's produced as a result of the analysis from the sample that was taken at the crime scene with the analysis of the individual that has provided the sample and is the suspect. And it's a question of, do these samples match? So what we have on this graph are a number of peaks and troughs. And what's significant and what's something to look out for in a sample like this is where there are 3 points. Because where there are more than 3 points, 3 alleles that have been identified, we know that what we're dealing with is a mixed sample as opposed to a sample from a sole person. We know that because when ordinarily one would expect to see 2 alleles, one from the mother, one from the father. When we see at least a third, we know that somebody else has been in with the sample. So the results that can be obtained, we can either get a full profile, which is when the DNA containing information on alleles within the marker system produces 17 full loci. We can have a partial profile, which is when not all 17 loci are present from within the sample. And we can, as we see in this particular graph, have a mixed sample where there are a number of different sources of DNA. Now, it is possible to say whether one person's sample is stronger than the other from the heights of the bars. The person with the weaker sample is going to have lower bars, whereas the greater sample is going to be higher up, indicating a stronger profile sample that's there. Can we move to the next slide, please, Andrew? So how do we establish the evidential significance of this match? Now, many of us will have come across this statistic, 1 in 1 billion, and a few of us will have questioned, well, what does that actually mean and how do we get there? Now, of course, the strength of the underlying DNA evidence is reflected in the value of the probability of a match that occurs between DNA profiles from the crime scene and that of the accused. The true position, of course, is that 1 in 1 billion isn't the actual and final statistic. That's simply a statistic that's agreed and used by the forensic science regulator because it's considered that it makes a point. It's significant enough so that everybody can draw its conclusion from it. But in order to calculate what that overall probability is, it's required to know what the relative frequency of a particular allele is occurring within a locus point within the general population. Now, when we calculate that probability, the relevant ethnicity of a person is taken into account, and ultimately when we multiply the various probabilities together, we get the final probability of an individual's DNA being matched by chance. Now, it's the forensic science regulator that oversees the frequency database that's used for DNA profiles. Now, that is different from the DNA database on which people who are convicted of recordable offences are entered into. And all forensic profiles, all forensic providers, when they perform these calculations, consider matters on the basis of 5 different ethnic groups: white, black, African or Caribbean, South Asian, Eastern Southeast Asian, or Middle Eastern and North African. But the number of samples within each category varies wildly, and perhaps the key document in relation to this is a 2014 paper by the forensic science regulator on allele frequency, but it varies from 2,550 allele samples within the white ethnicity to as low as 110 allele samples within the Middle Eastern or North African ethnicity. Now, it goes without saying, of course, that the quantification and that statistic of 1 in a billion is only as good as the underlying data that's used to produce it. But in assessing the quality of that available data, the 2014 document makes a comparison with a 2011 general population census. And it specifically states in the FSR's own document that the Indian population is underrepresented, and that the size of the data for Middle Eastern and North African individuals is far below than the 400 allele benchmark that it sets itself. So if your client is either of Indian ethnicity or Middle Eastern and North African, and they're involved in a case involving DNA evidence, well, the forensic science regulator's own documentation says that the sample size that they've got when coming to calculate these probabilities isn't particularly that good. So maybe that's something for cross-examination. Now, of course, work has been done over the past 6 years to try and ensure the databases are more reflective of the allele frequency in the population, and some statistical corrections have to be applied. Now, one type of statistical correction that's applied is within a local population where people may be more related to others than would be as a result of the population as a whole. I make no further comments on that. It may also be that the inclusion of a rare allele within a particular ethnicity overestimates its frequency, and so again a statistical bias has to be applied in respect of that. But in terms of how we calculate that figure, As the example on the slide shows, we look at the probability in respect of each particular allele frequency, multiply them together, and what we're left with is what are the chances that this particular DNA match that has occurred has happened by chance rather than anything else. Now, the premise behind the calculation and the premise behind being able to look at historical DNA data is something called the Hardy-Weinberg principle. Now, Hardy-Weinberg isn't a less famous cousin of Harvey Weinstein, but they both had run-ins with DNA. The Hardy-Weinberg principle states that the frequency of a particular allele within a population remains consistent over time. So if a crime took place many decades ago, the same genetic profile of the population in the present era is reflected in what it was back at the time that the crime was committed. And so it allows frequency databases like the present one to be used and for it to remain good evidence. But the Hardy-Weinberg principle does have a number of factors that cause it a few problems. It assumes that random mating has taken on, and we've already discussed briefly about how a statistical bias has to be applied to some areas of the country. It assumes that there's a large population size so that the probability of a particular locus can be readily established. It assumes that there's no mutation or migration, which is perhaps of more relevance to today's world, where people are moving between countries much more readily. And it assumes that there's no element of natural selection, so that no particular alleles have a productive reproductive advantage over others. Let's move on to the next slide, please, AJ. Right, Prosecutors' fallacy. The Prosecutors' fallacy is a fault in statistical reasoning. It's named after prosecutors, but it doesn't simply apply to them. Now, what the probability of 1 in a billion means is that the probability of a match occurring randomly. What it doesn't mean are the odds of a particular odds of guilt or innocence. The reverse of that probability and the reverse of the fallacy is that the probability represents the chances of somebody being innocent rather than guilty, and that's the defense fallacy. The fallacy, of course, has two separate limbs, the source probability error and the ultimate issue error. The source probability error suggesting that because of the DNA match, the DNA evidence establishes that the suspected individual is the source of the crime stain. And the ultimate issue error, because of the DNA match, the DNA evidence established that the suspected individual is guilty of the offence. That's not what that probability means, and that's the fallacy. So it's important, of course, that we don't make that error, certainly not when slipping up when making a closing speech. We're going to see an example of how that may work over the next two slides. So Andrew, can we move over to the next slide, please? Now, this was an example noted by John Keynes, playing poker against the Archbishop of Canterbury, and he draws himself a straight flush. Leads to two questions. What is the probability of him dealing a straight flush if he were playing honestly? And what is the probability of the Archbishop playing honestly, given that he has dealt himself a straight flush? Now, the prosecutor's fallacy would be to conflate those two questions and say that the probability of one is the probability of the second. That, I'm afraid, is simply not true, and we're going to see in the next slide shortly why that's not true. Now, mathematically, just under 2.6 million potential 5-card hands can be dealt with from a 52 pack of cards. So the probability of drawing a straight flush is about 1 in 72,000. But the second question, the probability of whether somebody is in fact playing honestly or not, isn't quite that clear, because of course there's factors that go beyond the maths. Somebody may well be of a religious nature and consider the Archbishop of Canterbury, of course, is playing honestly, and so the probability is closer to 1. The prosecutor's fallacy, as it says at the bottom of that slide, would be to argue that the probability of the Archbishop playing honestly, given that he's dealt himself a straight flush, is that mathematical probability. Now, if we slightly change the facts, we can see how the answer to the second question slightly changes and why the prosecutor's fallacy becomes an issue. And before we do that, can I just apologise to any Conservative voters who may be watching this. This individual has had quite a bit of coverage over the course of this webinar. And when I put this example together, I had no idea what Milo and Dennis were going to be talking about in their talk. So can we move to the next slide, please, Andrew? Rather than the Archbishop of Canterbury playing cards, you're now against Dominic Cummings on a trip to Barnard Castle. The question, the mathematical probability of the first question, of course, remains exactly the same because mathematically it's 1 in 72,000 of a flush being drawn. But the probability of the second question changes because the background facts have changed and our own views have come into it. And so it wouldn't be proper to draw the conclusion to the first question and apply it to the second, because we see how little things can make a difference to the probability. Next slide, please, Andrew. A couple of points just on limitations. Timing and means of placement of the sample. It's of course not possible to say when a DNA sample was left at a particular crime scene or how it came to be there. I'm sure we've all heard cries of movable item when speaking to a defendant about a stone or an item that was left at the scene of a burglary. Transfer of DNA, of course, transfer can take place in a number of ways: direct or primary transfer directly from the person that's left it. Secondary or even tertiary transfer can occur when a DNA sample moves between individuals. Elimination samples, we will want to know who has been eliminated before the expert's report has been finalised. So police officers have given their DNA over and their samples have been matched against that's been found and they've been eliminated. Potential for human error. Whilst a lot of this takes place involving chemical processes, computer interpretations, there are of course human beings involved at various stages through the DNA analysis. An error can of course creep in at some point. And finally, bone marrow transplants. It's worth inquiring whether your client has ever had one of these, because the effect of a bone marrow transplant is to introduce a donor's DNA into your client's DNA, which can cause elements of confusion. So that's an example of where DNA can in fact mutate. Almost final slide from me. Can DNA evidence alone be sufficient for a safe conviction? Well, this was first discussed back in '97 in the case of Doney, and Lord Justice Phillips made the comment. The stage may be reached when a match may be so comprehensive that it will be possible to construct a DNA profile that's unique and without anything else proves guilt of the defendant without any other evidence. Well, of course, you recall back in '97, there was the process of SGM, which only considered 4 separate locus points. We're far past that now, 23 years on, and technology has developed. It's not quite at the stage that Jurassic Park thought it was going to be, but maybe we'll get there in due course. The case of Jonathan Sakari provides a far more recent example of where this was considered and whether DNA evidence alone could be used to establish the guilt of an individual. Now, Mr. Sakari was charged with robbery. It was alleged that he'd opened the driver's seat door where the victim had been sitting, assaulted her and fled with a piece of her jewellery. And swabs had been taken from the door handle of the car, which came back showing that he was the major contributor to that DNA sample. Now, at his trial, he didn't give evidence and he was convicted, but the recorder during the course of the trial didn't allow a submission of no case to answer on the basis that was made on the basis that the sole evidence, in fact the only evidence, was that of Mr. Sakira's DNA on the car door handle. Well, the Court of Appeal disagreed with the appellant's submission and said that the DNA alone at the scene of a crime, without any obvious explanation, was sufficient to raise a case. There was no reason, there was no evidence adduced as to why his DNA would have been on the victim's car door handle. It did, however, note that circumstances could be present within a case. That means that the case could be kicked at half-time. Has an explanation already been provided in interview? That's something that the court said may well result in the case being removed at half-time. And so it's worth looking not only at the DNA, but also the inferences that can be drawn from the circumstances that are around it. Does the defendant have a reason for why he's in the location at the time? It may well be that something advanced at an early stage can counteract the presence of DNA. And Sakira says that that is something that may well result in a case being removed at half-time if there is nothing more. Final slide, please, Andrew. A shameless plug from my part. Book coming out in July 2020, a practical guide to the use of expert evidence, involving chapters on DNA, mental health, firearms, forensics, cell site fingerprint statistics, and a bit more. Website's available. If you have any questions, my email address is there. Thank you very much.